Good morning, everyone. Today I'll be discussing some of the leftover topics in embryology, putting into the title as miscellaneous topics in embryology. The competencies of today's topics are: describe the development and developmental basis of congenital anomalies of pituitary gland and eye. Describe teratogens, their role in congenital anomalies during first trimester of pregnancy. The contents in today's topic are development of the pituitary gland, development of eye, teratogens. Before studying the development of pituitary gland, let us see the gross appearance of the pituitary gland, which is fully developed. Here is the anterior aspect of the pituitary gland. Here is the posterior aspect of the pituitary gland. This is the superior and inferior aspects. It has two main parts called as adenohypophysis present anteriorly and neurohypophysis present posteriorly. Let us study adenohypophysis. It has the following parts. The bulkiest part of which is called as pars anterior, also called as anterior lobe. It is a narrow portion behind it called as pars intermedia. Also the part of adenohypophysis extending and towards the infundibulum is pars tuberalis. These are the three parts of adenohypophysis. I repeat, pars anterior, pars intermedia and pars tuberalis. The parts of neurohypophysis are the posterior lobe, also called as pars posterior, and its extension which communicates with the hypothalamus called as the infundibulum. The pituitary gland develops from two distinct sources. As we know, adenohypophysis, also called as anterior pituitary, it develops from the ectoderm lining the roof of stomodium. The posterior pituitary, also called as neurohypophysis, develops from the neuroectoderm of the hypothalamus. Observe this diagram. This is the superior or the cranial aspect of the developing embryo. This is the inferior, the caudal aspect. This is the dorsal aspect. Here is the ventral aspect of the developing embryo. This is the region where the primitive oral cavity or the stomodium is developing. The roof of the stomodium starts diverticulum, what you can see here as Rathke's pouch. So this evagination of the ectodermal lining of the roof of primitive oral cavity forms a pouch-like structure which is evaginating towards the developing brain. Is called as Rathke's pouch. It starts developing in the third week of intrauterine life. Later, this is cut off from the primitive mother stomodium and it joins the posterior pituitary. Now, let us understand this diagram. Here. This is the ectoderm that is lining the roof of the stomodium. Here is a diverticulum that is developing from the forebrain vesicle, which is future posterior pituitary gland. This is the anterior pituitary gland. Now let us see how they develop. In the diagram B, we can see this diverticulum of the ectoderm of the stomodium forms a pouch-like structure which is extending upwards. It is called as Rathke's pouch. Similar diverticulum from the forebrain vesicle descending down, we call it as neuroectodermal diverticulum. This will be lying posteriorly, this will be lying anteriorly. Then this Rathke's pouch gets completely cut off from the ectodermal lining of the oral cavity. But this diverticulum, which is from the forebrain vesicle, retains its attachment with the forebrain vesicle, and this forms the neurohypophysis, also called as the posterior pituitary. The Rathke pouch forms the adenohypophysis or anterior pituitary, which also gives rise to the three parts called as pars anterior, pars intermedia, and pars tuberalis. The neurohypophysis gives rise to the pars posterior and infundiculum of the pituitary gland. So these following five parts of the pituitary gland, three from the adenohypophysis, two from the neurohypophysis. The same thing being described here in a better picture. This is the developing oral cavity, and we call it as the stomodium. This is the ectodermal lining of it. Here is the diencephalon, especially the hypothalamic part of it. The neuroectoderm lining it is producing a diverticulum from above downwards. Here, the ectoderm of the stomodium is producing a diverticulum from below upwards. In diagram B, you can see here is what we call it as the Rathke's pouch, or also called as adenohypophyseal pouch. The diverticulum of the neuroectoderm, you can see, forming a neurohypophysis in the posterior aspect. The future, after the formation of the bone, that is the sphenoid bone, which has a fossa in it called as the pituitary fossa, is completely formed adenohypophysis from the ectoderm of the roof of the stomodium, which get completely detached. You can see this here. It has got detached completely and becomes adhered to the neurohypophysis to form a complete gland called as the pituitary gland. This canal, what you can see in the dotted line, it gets completely obliterated. The canal is called as craniopharyngeal canal. It's called as craniopharyngeal canal. It should completely disappear. Now this is what a completely developed pituitary gland looks like. We can see the adenohypophysis or anterior part of the pituitary gland, neurohypophysis or posterior pituitary. The anterior pituitary or adenohypophysis developed from the Rathke's pouch, which is the evagination from the ectoderm of stomodium. Thus given the pars anterior, pars intermedia, and pars tuberalis. Neurohypophysis as the pars posterior and infundiculum, which has developed as diverticulum from the diencephalon of the forebrain vesicle. It is the flow chart that describes the various parts of the pituitary gland as described in the previous diagram and slide. Now let us study the congenital anomalies of pituitary gland. Craniopharyngiomas. So you can see in this diagram, here you can observe properly, called as the tumor. We have studied the tract of Rathke's pouch forms what is called as craniopharyngeal canal. This craniopharyngeal canal should completely disappear. But if suppose it remains as a remnant, then it can develop into tumors called as craniopharyngiomas, and that are seen in the sphenoid bone. You can see in diagram D in a dotted line, this is called as craniopharyngeal canal. The formation of the pituitary gland completely, this craniopharyngeal canal completely obliterates. But if suppose it persists, then the tissue of the craniopharyngeal canal has a high tendency to form tumors called as craniopharyngiomas. And this is where we can see one such one called as craniopharyngioma. Now let us study the development of eye. We will describe the development of the eye under the following headings. They are development of optic cup and lens vesicle, and development of various parts of the eyeball. Please correlate this diagram with this. While the embryo is developing, this light green color here is nothing but the neural tube. You can see optic vesicle that has developed as a vagination from the developing neural tube. Now let us see this diagram. Here is the wall of forebrain, same thing here, and there is development of a sulci from this forebrain vesicle. You can see here completely developing. The sulci now is called as a vesicle. Now let us see how the optic cup develops. This is the forebrain vesicle, the wall of which you can observe here. That develops what is called as the optic sulcus. A pair of optic sulcus will be there. What in the diagram? For study, we have been shown only one. This is a cavity of forebrain. Let us see that this is the right sided one. There is also a left sided one that is supposed to form. This optic sulcus later on evaginates further to form the optic vesicle, and the narrow portion that is formed we call it as optic stalk. So the optic sulcus gives rise to optic stalk as well as the optic vesicle. We studied in the earlier slide how the neuroectoderm gives rise to the optic stalk and the optic vesicle. Now let us see the surface ectoderm that comes in contact with this optic vesicle. It develops an invagination called as the lens pit. The lens pit further invaginates deeper inside the developing optic vesicle, making it a cup-shaped structure. So this we call it as an optic cup now. This lens pit, it deepened due to further invagination, gets detached from the surface ectoderm to form a rounded structure called as the lens vesicle. Now we have optic cup here and we have lens vesicle. The optic cup form has two layers, the outer layer and an inner layer, which are continuous in the anterior aspect. The space in between the two layers of the optic cup we call it as intraretinal space. The space between the two layers of the optic cup we call it as intraretinal space. We will see what is the importance of this space in the later case. Try to understand these diagrams. It's important to understand what is the choroid fissure. Correlate with the earlier diagrams. We have seen what is optic stalk, what is optic cup, and the lens vesicle. From the inferior aspect of the developing optic stalk and cup, the mesoderm, invaginates into the stalk and the cup region from the inferior aspect and forms a choroid fissure. If suppose a transverse section of this is taken, you can observe here what it is like. Here is the optic stalk, inferior aspect of which is the choroid fissure. And this choroid fissure, which is nothing but the mesoderm, forms the blood vessels and the mesenchymatous tissue. Here we can observe that the choroid fissure, which was there carrying the mesoderm, has formed the hyaloid blood vessels called as the artery and the vein, which are also supplying the lens vesicle. In the earlier slides, we have studied the development of what is optic cup, what is lens vesicle, and what is choroid fissure. Now, further we study the development of individual structures of the eyeball. The first important one we study is the retina as well as the optic nerve. Let us see how the retina develops. We have studied from the four-time vesicle the development of the optic vesicle, which later on gets invaginated by the lens vesicle to form the optic cup. Once the optic cup is formed, it has an inner layer, outer layer, and an intraretinal space. The inner layer of the optic cup develops into a light sensitive neural layer of the retina. The outer layer of the optic cup develops into a pigment cell layer of the retina. So this is a nervous layer which helps in the vision. The pigment layer avoids the reflection of the light that enters into the eyeball. The optic stalk develops into the optic nerve. Having studied the development of retina and the optic nerve from the optic cup and the optic stalk, we will study the anomalies of the retina and optic nerve. What is retinal detachment? The retinal detachment refers to the separation of the pigments layer of the retina from the nerval or the neural layer of retina. This can be either congenital or acquired. We can also see what is a congenital papilledema. If suppose there is a bulged and edematous optic disc, now you may be wondering what is optic disc. The attachment point of the optic nerve to the eyeball is called as optic disc, as we see from the pupil. So if suppose this optic disc is enlarged and edematous, then we call it as papilledema. Since the optic nerve has coverings as that of the brain tissue, if suppose there is a raised intracranial tension because of the increase in the CSF, it is also shown in the eye as a bulged disc. We call that condition as papilledema. It can be congenital, it can also be acquired. Let us study the development of lens. The lens develops as a lens blockade of the surface ectoderm, which later on gets converted into a rounded lens vesicle, which has the lining of cuboidal cells. The lens vesicle later on forms an elliptical structure because of anteroposterior compression. Then the anterior portion or the layer of this lens vesicle has cuboidal cells, but the posterior layer forms tall columnar cells, which later on lose their nuclei and form fibers. We call it as lens fibers. The anterior layer forms an epithelial lining which is having cuboidal cells, but the posterior layer forms lens fibers once it loses its nucleus, and further the equatorial region cells keeps on forming the lens fibers and adding to the tissue of the lens. Here is what we can see. The equatorial region is the anterior aspect, here is the posterior aspect. This whole thing is the lens. These are the lens fibers. Here is the anterior layer. This is what here anterior. This you can see here. Then from the equator, you can see the cells that are developing, later on adding to the lens fibers. Now let us study the congenital anomalies related to the development of lens. You know that by the age of 50 or 60 years, people get what is called as opacity of lens. We call it as cataract. If suppose a newborn child has opacity of lens, we call it as congenital cataract. This usually happens when the mother gets infected during the pregnancy with torch group of organisms. Now let us see what are torch group of organisms. TO refers to toxoplasmosis infection, R for rubella infection, C for cytomegalovirus infection, and H for herpes infections. Any of these infections during the pregnancy can cause cataract of the lens of the newborn. We call it as congenital cataract. When you observe this diagram, through the pupil you are able to see the vascularity of the retina. This is because there is no lens C between the cornea and the retina. The absence of the lens is called as aphakia. If suppose it is absent in the newborn, we call it as congenital aphakia. So absence of the lens since birth, we call it as congenital aphakia. Now let us see what all components of the eyeball have developed from the embryonic structures. The embryonic structures that give rise to the components of the eyeball are mainly the neuroectoderm of the forebrain, the surface ectoderm in that region, as well as the mesoderm. From the neuroectoderm, we have seen how the optic cup and optic stalk are formed. The optic cup gives rise to retina. It also gives rise to the sphincter and dilator pupillae musculature of the iris. I repeat, the dilator and sphincter pupillae musculature of the iris. It also gives rise to epithelium of the iris and ciliary body. The optic stalk will give rise to the optic nerve. The surface ectoderm that forms the lens vesicle, which later on forms the lens. It also helps in the formation of anterior epithelium of cornea. The anterior epithelium of cornea. Now, what the mesoderm gives rise to, let us see. The sclera of the eyeball with the substantia propria and posterior epithelium of cornea and the choroid. These three entities develop due to the migration of neural crest cells into the mesoderm. The mesoderm by itself gives rise to the ciliary body, stroma of the iris, which means to say the connective tissue of the iris. Now see, the mesoderm gives rise to the stroma of the iris, but the musculature in the iris are developing from the neuroectoderm. Then the vitreous body developing from the mesoderm, the central artery and vein of retina, which we have seen as hyaloid artery and vein. When they lose a connection with the lens later on, they are getting converted into central artery and retina of the optic nerve. Then the mesoderm also gives rise to the muscles that surround the eyeball called as the extraocular muscles. Let us see other anomalies of the eye. Condition called as anophthalmia, which means to say absence of the eyeball. Microphthalmia is a small eyeball. Coloboma of iris is absence of the inferior portion of the iris. Cyclopia is presence of a single eye in the midline on the forehead, and proboscis refers to the presence of a tubular appendage which resembles the trunk of an elephant with the absence of nose in the area between the eye and the mouth. Now let us see this diagram. Here you can see the completely sunken region where there is no prominence of the eyeball. This eye doesn't have any eyeball inside. We call it as anophthalmia. It is because of failure of development of optic cup. Here it is partially sunken, which means to say there is eyeball which is smaller in size. We call it as microphthalmia. This is because of underdevelopment of the optic cup. Yeah, there is a deficiency of the development of iris in the inferior aspect. We call it as coloboma of iris. This is because there is failure of fusion of the choroid fissure. This is a case of cyclopia, where there is a single eye in the midline over the forehead, and there is no nose between the eye and the mouth, but there is a small tubular appendage seen on the forehead, which is called as proboscis. There is a separate branch of study called as teratology, which deals with the congenital defects and the causes behind it. And the mechanism of production of congenital anomaly in the developing fetus is called as teratogenesis. Teratology is the study, and teratogenesis is the process of the mechanism of development of congenital anomaly in the fetus. And the agents or the factors that cause these congenital anomalies, we call it as teratogens. For example, some of the infectious agents that can cause congenital anomalies, like we have seen, the congenital cataract. They are torch group of infections. The organisms are toxoplasmosis, rubella, cytomegalovirus, and herpes infection. Some females during pregnancy may also be exposed if they are working in factories that deal with chemicals like mercury and lead. They also have tendency of their kids being born with congenital anomalies. Suppose a pregnant female gets an X-ray done, which is ionizing radiation, then it may impact the baby that is developing in the womb of the mother. Some drugs which may be taken during pregnancy, like if suppose the female has excess of vomiting, nausea, or if she is epileptic and unknowingly she takes some tablets, like giving an example here, thalidomide, which is taken as a drug for reducing the nausea, which can cause congenital anomaly. Phenytoin, which was earlier taken as anti-epileptic drug, has tendency to cause congenital anomalies. And alcohol, the never-ending one. Okay, irrespective of if the female is pregnant or not, she consumes alcohol in affluent society. So there is high tendency of congenital anomalies for kids being born to such females. Now we should be knowing which is the susceptible period of pregnancy for teratogenesis. The initial 0 to 2 weeks of the pregnancy, we call it as period of morphogenesis. During this period, if suppose some abnormality develops, then it leads to the abortion called a spontaneous abortion may occur. So there is no question that a baby will be born with anomalies. From third to eighth week of pregnancy, we call it as period of organogenesis. This is the crucial period and most susceptible period for the teratogenesis. And if suppose the female is exposed to various teratogenic agents during this period, there is a high chance of the fetus being born with congenital anomalies. So this is the most sensitive period, and each organ system will also have a period of high sensitivity during this period if exposed to the teratogens. Beyond that, that is from 9th to 38 weeks of gestation, it is a less sensitive period. So this is a growth period. This is a period of organogenesis. This is the most susceptible period. So period of organogenesis is the one susceptible for teratogenicity. So every doctor should be aware of these following drugs which have a tendency to cause teratogenesis. So whenever you are treating a pregnant woman or whatever the clinical conditions she comes with, you should avoid these following drugs which have a high tendency to produce teratogenicity, the fetus that is developing within the womb of the female. These are the topics I am giving you, which you have to study and write down the answers for.
Under 5 marks questions, you have the following questions. Number one, alpha fetoprotein and its relation to the neural tube defects. Question number two, describe the indications, process, and disadvantages of amniocentesis. Third one, describe indications, process, and disadvantages of CVS, also called as chorionic villus sampling or biopsy. Fourth one, explain embryological basis of estimation of fetal age. The fifth one is ultrasonography in pregnancy. As a 2 marks question, you should also know what is pregnancy strip test. Thank you everyone.